Welcome back. Let's continue with our discussion of new genes. Dr. Man Yonglong had told you about some of his exciting research on new genes. In this unit, I will tell you about a surprising discovery of my own lab of a human-specific de novo protein coding new gene associated with addiction. You may remember from an earlier lecture that for a while my lab was studying drug addiction. We had a collaboration with a group of NIH NIDA, the National Institute of Drug Abuse in the US, to study their human genetic data on drug addiction. In particular, we analyzed several sets of genome-wide association data of drug addiction. Because most people focused on coding regions, we paid particular attention to SNPs in regulatory regions instead. Indeed, we found an SNP on the 3' UTR of a gene FLJ33706 that is statistically significant in 2 GWAS analyses of nicotine addiction and implicated in 2 linkage analyses. We looked at other SNPs in the nearby genomic regions, but found this SNP to be the most interesting. This SNP is located in the middle of 12 binding sites of microRNA LED7, which is known to play critical roles in brain function. And as you may know, microRNA binding sites tend to occur in tandem. So we were quite interested in it and started to think about experiments to test whether this SNP may change the binding of the microRNA. However, when we took a close look at the gene FLJ33706 itself, we found it to be very peculiar. There is no publication about it. Interpro found no detectable protein domain. There is no blast hit to any other known protein sequences in human or any other species. The UCSC genome browser and the NCBI untraced gene database even marked it as a non-coding RNA. However, when we looked at the gene closely, including its DNA sequences, available EST data, and its cDNAs that we sequenced, we found that it has a complete gene structure with 6 exons and a complete open reading frame encoded by exons 3 and 4. We used Pacman-based real-time PCR to find that FLJ33706 mRNA is enriched in human brain regions. We also used Western blot assay with an antibody designed against a 17 amino acid peptide to confirm the expression of FLJ33706 protein in human cortex, midbrain, and cerebellum. Immunohistochemistry studies of human cortex slides showed enrichment in cytoplasma of neuronal cells, so this is clearly a protein coding gene. What was really interesting and surprising was that through close inspection, we found that the gene is a human-specific protein coding gene that had originated de novo. This is a screenshot from the UCSC genome browser. Alignment of multiple vertebrates showed that the DNA segment emerged in eutherian mammals. The primates have similar DNA sequences as humans. If you look closely at the tree and the alignment, from the most distant primate species at the bottom to our closest primate species, chimpanzee and human at the top, you will see that 5 pairs of splicing sites gradually formed through the history of evolution. And chimpanzee and gorilla have exactly the same gene structure and splicing sites as human. What is most interesting is that only in human, 2 changes escaped 2 stop codons, making a complete open reading frame, enabling a 195 amino acid long human-specific de novo originated protein to be made. What about its upstream regulatory elements? If we look at the 5' region of the gene in the UCSC genome browser, we will see that data from the ENCODE project showed that there are signals of enhancers and transcription factor binding sites. Multiple sequence alignment showed that the promoter region is absent in chicken and zebrafish, first appears in mouse, and is similar in rhesus monkeys and chimpanzees. This pattern is similar to the pattern of the coding region. You may remember that the Neanderthal genome was sequenced. Did the gene emerge before or after the divergence of human and Neanderthal? Alignment of the Neanderthal genome to the human genome showed that the open reading frame also existed in Neanderthals. So the origination time of this protein coding gene was after the divergence of human from chimpanzee, but before the divergence of human from Neanderthals. The protein product has some interesting features. For example, it has a high isoelectric point of 11.8. Here we plotted the isoelectric points of all human proteins. As you can see, FLJ33706 isoelectric point is very high. We used COBUS to find which functional categories are enriched in human proteins with isoelectric point over 11. As the result here shows, among gene ontology functional categories with FDR corrected Q value less than or equal to 0.05, many of the categories involve RNAs and ribosome, such as RNA binding, RNA splicing, mRNA processing, mRNA metabolic process, etc. So if I had to guess, I'd guess that FLJ33706 may have functional roles involving RNAs. What would the three-dimensional structure of a protein that originated de novo look like? Would it have a completely different structure from other proteins? Or would it still fall into one of the known folds? As it was not possible to build a homology model because a de novo protein has no homologs or paralogs, we first predicted its secondary structure and found that it seems to have 4 helices and 1 beta strand. We then tried to use ab initio prediction software iTaser to predict its 3D structure. As you can see, different parameters gave completely different folds. We weren't able to get a reliable prediction. So this is still an open question. A lot remain unknown about this protein. Nevertheless, FLJ33706 was the first human-specific de novo protein coding gene associated with brain function. After we discovered FLJ33706, we asked 2 more interesting questions. How many other human-specific de novo genes are there? Where did they originate from? That is the subject of the next unit.